Well, I'm going to try to fuse the two ideas you just heard about, that is, doing genomic interrogation of a tumor with the idea of dissecting it on a single cell basis. And I'm going to give you a couple examples of things we've been working on in the lab recently, and I'll just sort of dive right into it. I think it's been set up pretty well by the previous speakers. The first thing I'm going to talk about is work that's been led by a fantastic fellow in the lab named Chuck Awad, along with, he's been collaborating with my student Winston Coe. When Chuck is not in the lab, he is actually treating pediatric ALL, which is the sort of most common pediatric cancer, and the cure rate is pretty good. It's 80%, but it's not perfect, and he's interested in trying to improve that. And one of the interesting things about ALL is that it's known to be a polyclonal disease, and people who have done sort of coarse measurements by CNV and looked at the dominant clone and diagnosis have been able to show that in cases of relapse, it tends to be quite a different disease. Only 8% of the time is it the same clone as it diagnoses as dominating the tumor. About a third of the time, it's some kind of evolution from that. Half the time, it's clearly evolved from some distant ancestral clone, and about 6% of the time, it just seems to be quite distinct. And so this is sort of very strong motivation to try to understand at a given point in time what the whole clonal structure of the tumor looks like, so that you can understand what the potential routes are to relapse and so forth. And so what Chuck has done has been to take actual patient samples and to try to dissect the clonal architecture. And we've done this using Fluidigm's C1 chip, which allows you to isolate single cells, amplify their genomes, and recover the full amplified genome from the chip for analysis. And this is the point where I should say, in full disclosure, that I'm sort of the scientific founder and consultant for Fluidigm. You could, at that point, with a lot of single-cell genome amplifications, just start sequencing them willy-nilly, and that would be very expensive to do. We've done it in a few cases of interest where there haven't been other approaches, but in the case of cancer, you've actually got a lot of information from the bulk tumor. So if you sequence the exome or the genome of the bulk tumor, that actually tells you what are the really interesting parts to look at of the tumor genome at the single-cell level. And so we tend to use that to focus in on what's interesting and go back and use those single-cell amplicons and do targeted resequencing there. And so from that set, that set of data, you then get, let's say, around 100 different regions of the genome that are interrogated per cell for single nucleotide variations. Chuck has also done CNV analysis and immunoglobulin recombination analysis. I'm not going to get into all those details. He's, I think, giving a poster here, and you can interrogate him about all that. I'm just going to give you a taste of what comes out from looking at the SNVs and what sort of the benefits you get from analyzing single cells over doing bulk analysis of allele frequencies. So this is what, for one particular patient, the bulk results look like, and you look at the allele frequency. So this is a histogram that's been turned on its side and reflected, so it looks like a violin. It looks very pretty. They're called violin plots. But just turn your head sideways, and it's a frequency distribution. And if this was a pure sample, you'd expect the minor allele frequencies to all be around 50% and 0. Here you can see there's a lot of intermediate values indicating that there's some kind of heterogeneity within the tumor and clonal structure. And people have worked hard to try to infer clonal structures from things like this, and you heard a bit about the approach that David is using at Santa Cruz. But there are limitations to that, as he noted, and one of them is it's very hard to look at clones of equal size. And in fact, here, when you look at the single-cell data, you can see that, in fact, there's quite a bit more to it. So here, each row is a different single cell. Each column is a location in the genome where an SNV is interrogated. Red is mutant, as determined from the bulk data, and white is the somatic genome. And so you can see that here there's clearly two clones, and they're quite different, and they're occurring with very comparable frequencies, so hard to infer that from bulk data. And you can start to build trees of what this thing looks like. In this case, it's pretty simple. There's some ancestral clone, and you can see these two dominant clones at the point of diagnosis. Here's another one. Again, the allele frequency from the bulk sample looks kind of like a big blob. There's clearly something interesting going on, but it's hard to infer what the clonal structure might look like from that. And here's what it looks like. So here you can see there's more clones, and they're quite distinct, and there's some relationships between them when you look at the sharing of mutations between the clones. And the tree ends up looking something like this. So you can see that the structure is a little bit more complicated and more substantial. And so it's sort of a really neat property that you can infer not only what the current distribution of clones look like, but also the history. I'm going to say a little bit about the technical details, just because there's not a lot of time. I didn't put a huge amount in about that. One of the challenging things in doing this and doing single-cell genomic analysis is your allele dropout rate. So it's hard to amplify the diploid genome and carry both alleles along. And so it ends up often limiting these measurements is what we call the allele dropout rate. Sites which you know are heterozygous, but because one allele drops out, they falsely appear to be homozygous. And we've worked really hard to characterize that for this approach by a number of different means. And it turns out to be about 20%, 25% allele dropout rate, which sounds horrible. I mean, it'd be very hard to do a whole genome-wide analysis with a rate like that. But in practice here, it's not so bad, and is manifested in these diagrams by the white spots in the middle of the clones. And so, if you look, around here, you can see that there's a non-significant dropout, a non-trivial dropout rate, but it doesn't impair you from determining the clonal structure. And the reason it doesn't impair you is that it's the large number of alleles you look at lets you overcome the dropout rate. And so if your goal is to determine clonal structure and dropout rate is the enemy, what your friend is the ability to look at lots of different locations in the genome, enough to overcome the dropout rate. And that can be quantified mathematically, and you can sort of formally work out one's confidence in determining a clone and the presence or absence of clones. And Winston did a lot of work in doing that, and again, I'm not going to dwell on it here, but there's a really nice sort of theoretical infrastructure behind all this, which we've been able to test with simulated data where we mimic the dropout rates. We put in a defined number of clones or a monolithic sample, and the algorithm does really quite well in sorting all that out. And so that's one of the nice things that's beneath the hood here. So with that in hand, we're in pretty good shape, and we feel that this is sort of just the beginning. Carlo, our moderator, and Mel Greaves has written, sort of nice articles about trying to use principles of evolution to infer what's going on in cancer, and with this ability to measure the actual clonal structure, we think the future is very bright for applying those ideas to these problems. Now I want to look at maybe a more challenging problem, look even further back in time. So if the clonal structure lets you infer the historical evolution of the tumor, I want to look back even earlier now and tell you a little story about what are the earliest mutations that occur that lead to the cancer. Really the very first ones, and presumably the causal ones. I'll describe some work we've done with Ravi Majeti, who's here at the meeting, along with his student Max Jan and my postdoc Tom Snyder, which we published the first chapter in a couple of years ago in Science Translation, and Ravi's gone on to use this approach very elegantly in other situations. And so here we want to look for the mutations that are present in the tumor that are fully penetrant. Okay, every cell in the tumor's got them, but they're not in most of the normal genome. But we're going to go back and look back at the very small population of normal cells, which has some of the mutations from the tumor. Okay, so those are the descendants of the very earliest origins of the cancer. And here we're going to switch to AML. And we use a similar approach philosophically, where the tumor was sequenced, the tumor exome was sequenced, interesting mutations that in this case were fully penetrant were characterized, validated by Sanger sequencing. And then from the same patients, where I was able to sort out residual normal hematopoietic stem cells, and we went and interrogated the genomes of those cells at the places of interest for the tumor. In this case, it's sort of a special example because HSCs can be grown into colonies, so we didn't need to do the microfluidic single-cell amplification. We just grew colonies that provide enough DNA to do the multiplex genotyping, but the whole philosophy is, again, sort of the same, being able to look at a number of different sites of interest in each single cell to work out the hierarchy. And let me give you a couple examples of that. So here are bulk data from 3 patients, and you can see for these different mutations, they're fully penetrant in the tumor, either heterozygously or homozygously, and yet that's shown in red. Blue is the normal bulk data from the normal hematopoietic stem cells, and you can see that these mutations are present in a small fraction of those stem cells. And the question is, what's their pattern of co-occurrence? Okay, from this sort of data, it's impossible to tell whether mutations even co-occur in different cells and what that pattern might be. But when you do the single-cell experiments, you can work that out. And here's an example of data from one patient where we looked at about 500 different cells, and the different axes here represent different mutations that always co-occurred, and each blob represents a single cell. And so the vast majority of the cells had none of these mutations. About 54 of them had SCIP2, so that would be, here's the vast majority with none. There's the 54 with SCIP2 on this axis. And then there were 3 cells that had SCIP2 and ELP2 and PTZD3, and there was 1 cell that had all of those mutations and CNDP1 and FLT3. And so when you think about what the Venn diagram looks like from this sort of data, it's clear that there was a single clonal history to this tumor in these normal cells, and the order of mutations is apparent. There's a concentric circle in the Venn diagram, and it must have been the case that SCIP2 was the first mutation to occur, and then in the SCIP2 mutated cells, some fraction also got ELP2 and PTZ3 and so forth and so on. And so this provides you a way to rigorously say, this is how it happened. This is the natural history of that tumor, and these were the very earliest events because we're measuring them in the residual normal cells. And so, very powerful approach to dissecting the natural history of a disease. In the remaining couple of minutes, let me just touch on a couple of other related topics. Returning to something that Gary mentioned, turning back to this idea of phenotyping the tumor rather than looking at the genotyping. RNA has been a powerful approach for us, and we've worked with Mike Clark's group and Irv Weissman's group for several years in doing this, and have been able to build similar sorts of trees of the phenotypic differences based on RNA patterns, initially with panels of genes, 50 genes, 100 genes, now whole transcriptomes, and that's a very powerful complement to this, to the genomic approach, and one that we think is going to begin to bear fruit, going to continue to bear fruit. And finally, I want to talk about epigenetics. That always comes up in these situations. If you can do single-cell gene expression, single-cell genome analysis, can you do single-cell epigenetic analysis? And the answer is that's a much harder thing to do, but there are some inroads that we've been able to make, particularly in collaboration with Bill Burkholder's group at IMCB Singapore, where he took one of the sort of classic methylation assays, which is to use a methylation sensitive restriction enzyme and PCR tricks around that site. And he managed to convert that and tune it up so that it worked on single cells, and was able to apply it in collaboration with Daniel Messerschmidt's group to problems in early development where there's mismethylation in mouse development. So the particular biology is not relevant to cancer, but I think the measurement is. And by looking at half a dozen different genes in each of the single blastomere cells, was able to work out patterns of mismethylation and look at co-occurrence and correlations. In this case, there's a lack of correlation. So when TRIM28 is knocked out, known to become sort of heterogeneously mismethylated in the embryos, and here he's able to show that the co-occurrence of mismethylation is consistent with completely random effects, so with a Poisson distribution. So in this case, you don't see correlations like we did in the previous example with the hematopoietic stem cells. But this sort of tool can also be brought to analyze methylation states in individual cells in tumors, and I think that'll happen in pretty short order. So I'll wrap up here and say that we're in a position to dissect genomic and transcriptomic diversity within individual cells. We've done it in a bunch of situations relevant to cancer with a number of collaborators here. And I'll stop here and take any questions, or we can go into discussion. Thank you.
Steve.
Yes.
In the last example, looking at one cell, two cell to blastocyst, it implies that perhaps at the blastocyst stage, not all cells in the inner cell mass are equal. They may look equal, but at the single cell level, those may not be mistakes in changing the methylation. They may be programmed changes within single cells, and so it would be worth going back in a system that you can study, like mouse blastocyst, to see to what degree of heterogeneity there is, and if any of the genetic changes predisposed to any particular germ line, mesoderm, ectoderm, endoderm, or tissue fate restriction.
I agree. Here it is bad to get these mismethylations, and it tends to be fatal. There's a fair amount of histology that was done of the embryos, but you can only take it so far. You'll be interested to know, he did a bunch of, Daniel, who's deep into this part of it, did a bunch of really interesting nuclear transplants between the knockouts and the normals, and a very interesting story behind that. So there's the functional thing that matters. Yep.
Okay.
Steve, excellent talk. You did a great job of describing the problem with the allelic dropout.
Mm-hmm.
I just had a technical question.
Mm-hmm.
Do you ignore the dropouts in constructing your trees, or how do you account for it? Do you just only count positive findings, or do you try to interpret, when you don't see a mutation when you construct
No,
your
we
lineages?
incorporate that knowledge, and so every cell we take the call as given, and we use the fact that there's a known non-zero dropout rate in making the trees.
So, okay.
Yep. And we've done it, a bunch of different ways, both by measuring the rate independently and putting that in as a fixed number, and also doing it in ways where we get the number out of the tree making, and it's all consistent. It's robust.
As compared to bulk sequencing, are you any more or less sensitive to the regional sampling problem, given that you're sequencing fewer cells, but obviously in a more detailed way?
I'm sorry, say it again?
Like if you repeat the experiment and sequence, single cells from one part of the tumor and then repeat it for another part of the tumor, do you expect that that would be more or less robust to that sort of gross level of heterogeneity as opposed to bulk sequencing?
Well, these are liquid tumors, so you don't get the geometric part of it. I mean, it's all going to be transferred over to solid tumors, and these questions about regions are going to be pretty interesting to sort out, I think. And as you know, there's been a literature on people doing bulk sequencing on subsections that are sort of chosen from different geometries, and one can see differences there.
Yeah, but you don't have a sense of whether this will be more or less sensitive?
I think it depends on the tumor.
Yeah. Okay.
It'll depend on the question you're asking.
What % of normal hematopoietic stem cells have these pre-leukemic mutations, and what % of patients with the mutations, pre-leukemic mutations, actually go on to develop the leukemia?
So, it's on the order of a few percent, and it varies, some more prevalent than others, but, single-digit percentages of hematopoietic stem cells seem to have these mutations. And 100% of patients had leukemia. So this is a look back in time. So these are people who are sick with leukemia, and we're looking at the residual normal hematopoietic stem cells while they're sick, and these are the descendants of things that made the
I'm asking,
tumor.
can you use this for a screening to look forward in time?
We haven't done that yet, but it's a good question. Yeah.
Great. All right. Let me have the speakers come up to the panel, and we'll have a more open discussion about the issues around heterogeneity and measurement.